Good morning, children. How are you all? I hope you each and every student are fit and fine. So, children, today we are going to start a new topic, the processes of recombinant DNA technology. Yesterday we have discussed about the recombinant DNA technology and then the principles of biotechnology. So children, this is the topic of chapter 11, Biotechnology Principles and Processes. So there are the various steps of the recombinant DNA technology. In this process, the first step is the isolation of the genetic material, which is DNA. Then cutting of the DNA at the specific location. After that, the amplification of the gene of the interest using the PCR. PCR means polymerase chain reaction. After that, the insertion of the recombinant DNA into the host cell, and then the obtaining the foreign gene product. And the last process is the downstream processing. So children, the first is the isolation, in which they treat the bacterial cell or the plant or the animal tissues with the enzymes like the lysozyme in the bacteria, cellulase in the plants and the chitinase in the fungus. The cell is broken and releasing the DNA and the other macromolecules like RNA, proteins, polysaccharides and the lipids. So children, in the isolation of the genetic material, RNA is also removed by treating with the ribonuclease, and the proteins are removed by treating with protease enzyme. So the other molecules are removed by the appropriate treatments. When the chilled ethanol is added, the purified DNA precipitates out as the fine threads in the suspension. These techniques generally we are using in our lab for the extraction or the isolation of the DNA from the plant material like banana, spinach leaves. We are using these materials. So children, the second process is the cutting of the DNA at the specific location. The purified DNA is incubated with the restriction enzyme. As a result, the DNA digests and the DNA fragments are separated by a technique called the gel electrophoresis. And the agarose gel electrophoresis is employed to check the progression of the restriction enzyme digestion. So children, in this picture you can see the DNA bands. Some small bands and some largest bands are developed in the gel electrophoresis technique. So children, the DNA is the negatively charged and we know it moves towards the anode. So the DNA fragments are separated according to their size through the sieving effect of the agarose gel, and the smaller size fragments move farther. So the process is repeated with the vector DNA also. Then the DNA fragments can be seen as a bright orange colored band when they are stained with the ethidium bromide and exposed to the UV radiation. So children, the DNA bands are cut down from the agarose gel. This is called the elution process. And after the elution process, the cut out gene of the interest from the source DNA and the cut vector are mixed. And then the ligase is added because the ligase enzyme is used for the joining process and it creates the recombinant DNA. You can see in this picture the recombinant DNA, children. Yes. So after that, the third process is the amplification of the gene of interest using the PCR. PCR means polymerase chain reaction. Is the synthesis of the multiple copies of the gene of interest in the in vitro using the two steps of the primers and the enzyme DNA polymerase. So the primers are small chemically synthesized oligonucleotides that are complementary to the region of the DNA. So children, the amplification of the gene of interest using the PCR having three main steps. The first step is the denaturing, and the second is the annealing, and the third is extension. One by one, now we are going to study these processes. The first is the denaturation. It is the heating of the target DNA or the gene of interest at the high temperature according to 94 degree Celsius to separate the strands, and each strands act as a template for the DNA synthesis. After that, the second process, in which it is the joining of the two primers at the third end of the DNA templates, which is the annealing. And the after that, the second process, annealing, the third is the extension. Extension is the adding of the nucleotides to the primer using the thermostable DNA polymerase called the tag polymerase, and it is isolated from a bacterium Thermus aquaticus. So children, it remains active in the high temperature during the denaturation of the double-stranded DNA. And through the continuous replication process, the DNA segment is amplified up to 1 billion copies, and the amplified fragment can be used to ligate with the vector for the further cloning process. So, children, after that, the amplification of the gene of interest, the denaturation process, annealing process, and the extension, these three processes. And then the fourth is the insertion of the recombinant DNA into the host cell. Using the any method, the ligated DNA is introduced into the recipient cells, or we can say the host cell. They take up the DNA from its surrounding, and if a recombinant DNA bearing the ampicillin resistant gene is transferred in the Escherichia coli cells, the host cells become ampicillin resistant cell. And if the transformed cell are spread on the agar plates containing the ampicillin, only the transformants will grow, and the untransformed recipient cells will die because they are resistant. And after that, the fifth process is the obtaining the foreign gene product. The ultimate aim of the recombinant DNA technology is to produce a desirable protein. So, if a protein encoding foreign gene is expressed in a heterologous host, it is also called the recombinant protein. So children, the cells with the foreign genes can be grown in the laboratory, and the cultures are used to extract the desired protein and purify it by using the separate techniques. The cells can also be multiplied in a continuous culture system. Here the used medium is drained out one side while the fresh medium is added from the other side. It maintains the cells more physiologically active and so produces a large biomass. So it yields more desired proteins. So children, these are the bioreactors. These are vessels in which the raw materials are biologically converted into the specific products, enzymes, using the microbial plants, animals, or the human cells. The bioreactors are used to produce the large quantities of the product, and they can process 100 to 1000 liters of the culture at one time. The bioreactors provides the optimal growth conditions like the temperature, pH, the substrate, salts, and the vitamins, oxygen to get the desired product. So children, these are the bioreactors which are mostly used. The bioreactors, the example is the stirring type, stirrer tank bioreactor. It is usually cylindrical in nature and with a curved base to facilitate the mixing of the reactor's contents. The stirrer facilitates even the mixing, or we can say the oxygen availability. Alternatively, the air can be bubbled through the reactors. So children, the parts of the bioreactor is an agitator system, oxygen delivery system, foam control system, temperature control, pH control, sampling ports. It is a very important topic because generally the examiner asks the question from the topic bioreactors and draw a well labeled diagram of the bioreactors. It is very important, so learn it carefully. And the last process of the recombinant DNA technology is the downstream processing. It is a series of the process such as the separation and the purification of the products after the biosynthetic stage. The product is formulated with the suitable preservatives such as the formulation undergoes clinical trials and the strict quality control testing. So hope children this topic is clear. Any query you can write in query box. Thank you children.